Hi, this is a recording. My name is Max Mekish of Rempel, and I will speak today about molecular structure considerations for sequence-dependent DNA resonances. And my company is dnaresonance.org. I will start with a chronology of the biofield research. The term biofield encompasses electromagnetic and acoustic fields regulating living systems, excluding biochemical, neuronal, and mechanical signaling. I'll start with ancient mentions of the biofield, prana in ancient yoga, chi energy in ancient traditional Chinese medicine, healing energy in many ancient cultures. The scientific research starts with Franz Mesmer in 1774, who demonstrated animal magnetism in experiments with people. He called it animal magnetism. In 1810, Berzelius introduced vitalism and defined it as that life is driven by a vital force. In 1840, Johannes Peter Müller said that light and sound waves show that living organisms possess a life energy unexplainable by physical laws. 1892, Hans Drake proposed biofield experiments with fertilized urchin eggs. 1920s, Alexander Gurevich, Hans Spemann, and Paul Weiss independently proposed morphogenetic field. And morphogenetic field is defined as a field which determines the shape of the body and the structure of the organs. 1920s and 1940s, Alexander Gurevich produced experimental evidence for mitogenic radiations. The idea of DNA resonance is that DNA sequences send and receive electromagnetic waves, guiding the morphogenesis of the organism and the functions of its cells. It's not the biofield fact, but it's essential for the story. In 1953, the DNA double helix was discovered by Watson, Crick, Wilkins, and Franklin. In 1960, experimentally, Lev Blumenfeld discovered magnetic properties of the DNA. In 1962, Boris Deryagin discovered polywater experimentally. In 1963, Per Olof Lodin proposed quantum genetics and proton tunneling and delocalized pi electrons in DNA. 1968, Hermann Froelich proposed long-range coherence and cellular resonances. 1969, Stanford Goldman proposed biological quantum mechanics and DNA hologram. 1972, Richard Alan Miller expanded the idea of DNA hologram and Bort web and proposed that DNA creates biofield. 1978, Fritz Albert Popp introduced experimental biophotonics, and in 1984 proposed that DNA is the source of the biofield. 1987, Rupert Sheldrake proposed that DNA is an antenna for morphic field and introduced morphic resonance. In 1988, Jacques Benveniste published water memory experiments. In 1994, Peter Garyaev published wave genome idea. In 1994, Stuart Hameroff and Roger Penrose proposed information transfer via microtubules and a quantum model of consciousness. In 1999, Alexander Burlakov demonstrated morphogenic field experimentally using fish embryos, and in 2017 demonstrated the effects of DNA fields. In 1999, Michael Levin showed experimental effects of static electric fields in morphogenesis. In 2003, Gerald Pollack published experimental easy water properties, which is essentially polywater, and later radiant heat affecting water structure. And he also later proposed that DNA helix shapes water layers. In 2009, Luc Montagnier published kilohertz resonance electromagnetic frequencies of DNA solutions. In 2012, Konstantin Mail proposed structured electromagnetic waves for DNA resonance. In 2018, Philip Ball proposed that radiant and diffusion heat transfers biological information via quantum entanglement. Let's now focus on water structure near DNA. As I said, in 1962, Boris Deryagin discovered polywater, a structured water formed near surfaces. In 1969, Lippincott proposed the polymeric structure for polywater. It branches, and to keep it neutral, the branches cannot reconnect. And this causes the branches to be separated by gaps or canyons. And this also should cause the continuous dynamic restructuring of this polymer. In 2013 and 2013, Gerald Pollack studied polywater extensively. He called it easy water. He proposed that polywater forms around DNA, that its layers are perpendicular to DNA, and that every layer is shifted stepwise by 1.5 angstrom in one of three directions, and that DNA imprints its helical structure on polywater via these shifts. In 2023, we proposed that there are two water layers, polywater layers, per every base pair in DNA. Since it is B form, base pairs are shifted depending on the sequence, and the shifts are imprinted on the polywater structure. Therefore, we propose the mechanism of imprinting DNA sequence on water structure. And here you can see identical DNA double helices, two, and these layers are shifted in three directions depending on the DNA sequence. So the structure proposed is published in 2023, and that's the reference to the structure. You can find the details of the structure. And it is in agreement with the reports on sequence-specific DNA resonance in solutions. And here are the reports by Edwards, Ichloff, Goodman, and Montagnier. And two of the reports describe microwave DNA resonances, one extra low frequencies and radio frequencies, and Montagnier describes kilohertz DNA resonances. How is it possible to test this model experimentally? An obvious, straightforward way is the micro-C method. It takes a snapshot of chromatin folding by first cross-linking the DNA and proteins, producing a soft polymer, breaking the materials into small pieces, cutting the unprotected DNA ends with micrococcal nuclease, ligating the sticking out DNA ends to each other, and sequencing the ligated pairs. We use the experimental results produced by the method by Hsaich 2016. So in this data, we know which pairs of sequences adhere to each other. So we tested whether these pairs are more homologous to each other than to the sequences over 1 kilobase away. So we take the sequence, search for homologs in purine code with the parameters that they should be longer than 15 base pairs and should contain less than 3% mismatches and should have no gaps. As a result, we see that the sequences that touch each other contain many more homologs in each chromosome than the sequences that are 1 kilobase away from the ligation point. So this confirms that the homologs are actually present in the sticking sequences more frequently. So the experimental micro-C sequence data confirmed our prediction with a high statistical significance. It is well known how complementary single-stranded DNA strands stick to each other. In the proposed sequence-specific homologous double-stranded adhesion. It is different since it works through structured polywater layers. So there are two double-stranded DNAs connected through structured polywater layers. Here are interesting consequences of that. Double helixes with similar sequences should stick to each other, leading to sequence-specific chromatin folding. And chromatin folding is well studied and it is important. Also, 55% of the genome are repetitive transposons with millions of copies. The role of transposons is to provide sticky points to fold chromatin. And the next consequence is that dynamic chromatin restriction is how the cell thinks. This is similar to neural nets. Another interesting consequences is that DNA solutions in living cells have two reasons to constantly restructure. The structure of polywater is imperfect. As a honeycomb crystal grows, it gains charge, and to keep itself neutral, the structure grows on one end and dissolves on another end. The symmetries of DNA in water are different. Polywater and nucleobases have hexagonal symmetries, while DNA in B form has 34.3 degree symmetry. This should result in a partly unstable embedding of DNA in polywater structure. DNA likely constantly twists and alternates which base pairs fit in a polywater structure. This doubly unstable DNA-water structure leads to constant chromatin reorganization and is the driving force of life. Soon after the discovery of the double helix in 53, Blumenfeld discovered that DNA can be magnetic, 60. DNA can magnetize temporarily like an electromagnet because its aromatic rings and base stack can spin collectively, producing ring currents. This is why DNA turns perpendicular to the magnetic field. It is known that magnetic fields repress cell cycle. Temporary magnetization of DNA can serve a number of functions. DNA strands could attract and repel each other using magnetic force. Since DNA is negatively charged, the magnetic field in the core of DNA can cause a number of things. Winding of the DNA double helix on the S magnetic pole and unwinding on the north magnetic pole. Magnetic DNA could propel itself through the water by screwing into it. Magnetic field produced by DNA could create vortexes in a nucleoplasm containing ions. Nucleoplasm is a heavy fluid containing 1% of DNA and 20% of proteins containing aromatic and charged amino acids. Magnetization of DNA wrapped around the nucleosomes could cause a histone core to spin. So histone core would spin. What can cause the temporary magnetization of DNA? While DNA is negatively charged, the histone core is positively charged. And spinning of the histone core within DNA loops or sliding DNA through the histone core should cause the ring currents in aromatic rings of the base stack, according to the Faraday induction. And these currents will temporarily make DNA magnetic. In addition to temporary magnetism, DNA also is a conductor of electricity. The double helix contains 3 wires, 2 isolated wires and 1 ground wire connected to water. The 2 isolated wires are the stacks of fused aromatic pi rings of nucleobases in each of the DNA strands. The 1 ground wire is made of longitudinal hydrogen bonds that connect the bases on the outside of the base stack. So wire 1, wire 2, and wire 3. Again, in a vertical model of DNA, wire 1, wire 2, and wire 3. What can cause electric currents in DNA? Spinning of the histone core of the nucleosome can. So if the histone core is spinning and it is charged, then it will induce electric current in the DNA. Even non-biological microscopic movements of water, lipids, and detergents are organized in vortexes, clouds, blobs, ridges, spins, periodic patterns, and fractals. We suggest that chromatin does all of the above, plus much more, since it contains DNA, histones, and other proteins. The nucleus is a sophisticated factory. How is the structure created? And how the molecules propelled to their positions? Here are suggested mechanisms. In previous slides, we described how DNA sequence structure propagates through water as a dynamic liquid crystal. Chromatin dynamically reorganizes via self-homology, via the perpendicular layers of water in a sequence-specific manner. DNA loops attract or repel each other magnetically. DNA propels itself by screwing into water. DNA controls the twisting and untwisting magnetically. Chromatin organizes via cymatics by creating and gradually adjusting standing ultrasound waves. Electric vibrations are tied to acoustic vibrations via electroacoustic principle. Electromagnetic waves applied to charged macromolecules result in ultrasound, and ultrasound applied to charged macromolecules results in electromagnetic waves. Chaos theory can model chromatin restructuring. Percolation theory can model how the complex network makes and undergoes such large transformations as chromatin rearrangements, folding and unfolding for gene expression and cell division. Acoustic and electromagnetic vibrations go through DNA strands. They also go between DNA strands via the liquid crystal. Nucleosomes help to structure electroacoustic sequence interactions. Although understanding chromatin remodeling in all complexity might be difficult, there is a goal that is attainable in the short term. This is to decipher the vibrational code of the genome. Only 4.5% of the genome code for protein. So we believe that 95% function as a vibrational code for morphogenetic field. Especially promising is the repetitive part of the genome, which is 55%. The vibrational function of the repetitive DNA is quite different from classical genes, since vibrational patterns should be analyzed collectively via chaos and percolation models. Yet, with emerging improvements of artificial intelligence and large language models, it becomes possible to model and decipher the vibrational code of the genome quickly. The vibrational genomic code will be useful in many fields. Vibrational code will explain the function of millions of non-coding mutations. Vibrational code will help to develop vibrational medicine, and that would solve the task of organ engineering and regeneration. And the vibrational code will help to understand how our mind works and speak to it in a vibrational language. In conclusion, I would like to thank my current and past co-authors. This work was funded by me in a small part by crowdfunding, and I'm thanking my sponsors. All the publications in PDFs and video presentations you can find on my site dnaresonance.org, and here are my email and phone contacts. And invite questions, please send them to me.